Hi, everyone. Thank you for the introduction. My name is Phoebe. I am a PhD student in Dr. Amy Ramsey's lab at the University of Toronto. My presentation title for today is Characterizing Mitochondrial Function and Morphology in Mice with Missense GRINI patient variants. So to talk about GRINI genes, I need to first give everyone a recap of an important receptor in our brain, the NMDA receptor. It is an ionotropic receptor activated by glutamate, the major excitatory neurotransmitter in our brain. So a functional NMDA receptor is a heterometric complex, consists of 4 subunits, 2 of which are GRINI subunits, which is present in all NMDA receptors. The other 2 is a combination of any of the 4 GRINI2 or 2 GRINI3 subunits. So NMDA receptors are cation channels that mediate slow inward current and calcium signaling at the synapses. Therefore, they play an important role in brain development, synaptic plasticity, learning, and memory. Although its function is most well characterized at the synapse, it's also worth mentioning that they're also expressed extrasynaptically, where they're believed to play an important role in excitotoxicity. They're also expressed in cells other than neurons and even outside the brain. So NMDA receptor dysfunction is associated with many neurological conditions. More recently, a group of patients first diagnosed with different neurodevelopmental or psychiatric disorders were found to have a mutation in one of the 7 GRINI genes that encodes the NMDA receptor. So patients with this rare genetic condition has a spectrum of severe phenotypes, including intellectual disability, developmental delay, autism-like spectrum disorder, seizures, movement, epilepsy, visual cortical impairments, movement disorders, and so on. So we realized that some patients with mitochondrial diseases that affecting their central nervous system function actually have similar symptoms, such as the learning disability, developmental delay, movement disorders, and so on. Therefore, we wondered if some of the phenotypes we saw in patients with GRINI disorder are actually due to mitochondrial dysfunctions in these patients. So another evidence linking NMDA receptor to mitochondrial function is that a group showed in 2011 that NMDA-like receptors may be actually present on the mitochondria. And the same group published last year showing that those receptors may actually contribute to mitochondrial bioenergetics. So we have several mouse models to study GRINI disorder. The first is the GRINI knockdown mouse, which has a global homozygous loss of function mutation in the GRINI gene that leads to a 90% reduction in GLUN1 expression. Therefore, it's a great model for studying NMDA receptor hypofunction with phenomenal phenotypes. More recently, in collaboration with Dr. Bill Pavin at NIH, we also generated mouse lines that possess the same missense heterozygous variants in patients with GRINI disorder. So the 2 I'm going to use in my project are GRINI1 Y647S, which has a gain of function mutation in the pore-forming domain of GLUN1. The other is GRINI1 Q536R, which has a loss of function mutation at the glycine binding site of GLUN1. So I will show you a little bit more evidence discovered in the GRINI knockdown mice, which supports why we think mitochondrial bioenergetic defects might actually be present in GRINI disorder. So Wesley et al. Published in 2014, where they a proteomic analysis paper on the GRINI knockdown mice, where they found proteins in the glycolysis and TCA cycle, namely the pyruvate kinase, metal aspartate, and aminotransferase were actually overexpressed in the frontal cortex and hippocampus of GRINI knockdown mice. In support with that, several papers have shown that there is excessive firing of neurons in different brain regions of the GRINI knockdown mice. So showing that they might have increased mitochondrial respiration, and it seems like they do have higher energy demand to support their firings. Inconsistent with that, our lab also have some preliminary data using the seahorse analyzer, where we showed that the primary cortical cells isolated from postnatal GRINI knockdown mice have shown elevated oxygen consumption rate, mitochondrial basal respiration, and ATP production. So it's also supporting the high energy demand in these mice. But at the same time, the same paper showed by Wethling and supported by other papers shows that there is also reduced glucose transporter expression in the brain of GRINI knockdown mice. And there is also another paper, although not shown here, show that there's reduced glucose uptake into the brain of GRINI knockdown mice. So even though they have high energy demand, they're not actually getting enough glucose substrates. So we don't really know what is going on there, but altogether, there's several pieces of data suggesting that there is bioenergetic failure present in GRINI knockdown mice, and it's involving the mitochondrial function. Therefore, it really made us wonder if the mitochondrial function is also altered in the mice with patient variants, which could be more clinically relevant. So therefore, it leads to my projects. There's two objectives in my project. The first is to characterize the mitochondrial respiration in neurons and astrocytes dissociate from the mice with GRINI patient variants. We hypothesized that GRINI variants will have increased mitochondrial respiration in their neurons and astrocytes. The second objective is to look at the number and morphology of GRINI of mitochondria in the GRINI patient variant mouse lines. And we can do that with transmission electron microscopy. We hypothesize that the bioenergetic failure will be evident by increased number of mitochondria and altered morphology of mitochondria in the axons of the corpus callosum. To achieve my first objective, we will use the gentle max dissociator by MITO2i Biotech, which really allows us to apply gentle mechanical dissociation so that we can get viable cells from adult brain tissues instead of postnatal. We will then use the max separator to isolate neurons and astrocytes, which will be plated separately on 24-well or 96-well plates, seahorse plates coated with PDL. We'll grow the culture for 5 to 7 days and make sure the cells are healthy before we move into the mito stress test using seahorse XF analyzer, which is the same test we used before for the GRINI knockdown cells. So to achieve my second objective, we will fix the adult brain tissue containing the corpus callosum region and prepare the samples for TEM imaging to visualize and characterize the mitochondrials in the axons. So although I don't really have any data to share today at this but I can share a little bit of our patient engagement strategies here. So our lab works very closely with the families of the patients carrying the GRINI variants. Actually, the family members approached us and leads to the generation of the patient variant mouse lines we have and will be used in my project. We work closely with them to understand their concerns, which actually helped us better design our study approaches. In addition to that, our lab also works very closely with GRINI, which is a parent-organized foundation that connects the families with researchers and clinicians in GRINI disorder. So my PI, Dr. Amy Ramsey, is actually a member of their scientific advisory board for GRINI. They also hold annual conference and monthly roundtable discussions. Our lab contributes regularly to these two events, which is not only an opportunity for us to share our data to the patients, but also a good chance to hear the feedbacks from the patients, which helps us to adjust our study approaches. And lastly, we'll collaborate with clinicians for translational potential of our discoveries and may lead to N-of-1 clinical trials in the patients. And with that, I would like to thank everyone in the Ramsey Salapur lab and all of our funding sources, and especially MITO2i for rewarding me and having me in this great event. So thank you.